Hello everyone. So in this session we will discuss about regulation of gene expression. We have talked about the replication and transcription beforehand. So and then we also saw how the mRNA which is synthesized by transcription, how they can be processed by 5' cap addition and neutron splicing and all. So and I think in the last session we talked about the protein metabolism, translation we discussed and its regulation. So transcription we have already discussed, which is the process of expressing genes. So now we have to see that what are the regulatory factors which can modulate or which can control the gene expression or the transcription process, because it has to be very tightly regulated. In our genome, there are different sections are there. For example, euchromatin and heterochromatin. So the reason that some portion of our genome is euchromatin and some are heterochromatin, the reason is the tight regulation, right? The heterochromatin regions are more accessible to the transcriptional accessories, RNA polymerase, and other factors. That's why they are very actively transcribed region. But if we talk about the heterochromatin region, for example, ribosomal DNA region, right? Or the telomeric region, they are the heterochromatin region. So they are not allowing the a good access of the RNA transcriptional machinery, right? So that entire process is called as regulation of gene expression. So now we will see what are the factors, what are the process which which are playing role in this important method which can regulate the transcription process. So first we have seen that initially that from genome, particular gene can be transcribed by the process of transcription, and then the primary RNA transcript can be processed, can be matured, right? And right, among them 5' G capping is a uh, processing, right? Intern splicing is a processing. So after all of this processing, there will be a mature messenger RNA, right? So now this messenger RNA will be used as a template for the protein synthesis by ribosome. So first from the nucleus, this entire transcription process and the processing process that occurs inside the nucleus, but when it will synthesize a matured mRNA, that will be exported to cytosol from the nucleus. And in the cytosol, it will get access to the ribosome, right? That we discussed in our last session. That ribosome has ribosomal RNA and some ribosomal proteins. They have two subunits, small and large. So this ribosomal assembly will initiate the process of the translation when it will get the access of this mature messenger RNA, right? And then there will be after when the protein will be synthesized here, right? So this is the inactive protein. So now in eukaryotes specifically, the the synthesized protein has to be undergone some post-translational processing. For example, glycosylation, right? So that and of course phosphorylation, and it has to enter into the endoplasmic reticulum so that it can be folded properly. Some proteins doesn't need to require to transfer get into the ER, but right? In this, by the help of the cytosolic chaperone only, they can be uh, folded properly. But some proteins needs to acquire some extra folding, so that's why they need to go to the endoplasmic reticulum, right? So there they will be further folded, and some processing will also be there. For example, addition of the glycosylation group, right? So now after all those processing uh, processes, the proteins will be modified and it will be active, right? And then the protein will be targeted to its right, so that it can be localized to the proper organelles, right? Or if it is a protein which has to be exported, then it will go to the cell membrane from where it will be exported to the outside. So this is the main, the steps for our central dogma, which starts from the DNA or our genome. The genes are getting transcribed to RNA, from RNA to protein, and then protein undergoes some processing, right? Then you will get a modified and active protein, right? So in this process, this transcription, right, here, this transcription process, so this process is very tightly regulated. Some genes are lying in the heterochromatic region, so their transcription rate is very low, and some genes are in the euchromatic region, so that their transcription rate is very high. So now how this difference can be maintained, can be regulated, this expression, that we will see. So first of all, as we as we know, if we you know uh, again revisit the entire process of the transcription, then we have seen that we started from promoter region, right? In eukaryote and as well as in prokaryote, the transcription process is controlled by the upstream region, which is called promoter, right? So if you see the construct for a E. coli prokaryotic promoter, that is the upstream promoter element, UP element, which is distant from the RNS start site, which is followed by a minus 35 region, which is the composition is TTGSEA, and there is a minus 10 region, which is TATA80, which is also called as Tata box, right? So in this, so this minus 35 and minus 10, they are lying inside the promoter region, and where the RNA polymerase will come and it will bind, right? Yeah. So now this is the consensus region for the promoter minus 35 and minus 10, but there are some other regions also which is not defined, right? So that can be different for different genes. Yeah. So that can be different for the different genes. So, and there is also UP element, right, which is also distant from the promoter, and it is also, there is no consensus sequence, right? So it can be different for different genes. So this entire composition will actually, of course, play a role in regulating transcription, right? But for E. coli, there is no such, chromosome, I mean chromatin, right? Although there are some histone-like proteins which are actually helps the DNA to be folded, but in eukaryote actually we will see that in the chromatin there is chromosome as well as the histone protein, right? Which helps the chromosome to be folded properly, packed properly, right? So that actually plays a role. Uh, this histone protein and this upstream promoter element composition, right? Promoter composition, they actually play a role. I mean, so both the upstream element of the gene as well as the histone protein, so both of them will play a role to regulate the transcription process, right? So this is consensus sequence of for promoter that regulate expression of the E. coli heat shock genes. So heat shock genes, so their expression is not constitutive. So normally they will not be expressed. But when you will give heat shock to the bacteria, right? For example, 37, I mean, for example, 42 degrees Celsius, then the bacteria will start to express those heat shock proteins by heat shock genes. So that means this heat shock gene transcription is properly regulated, right? Strictly regulated. Only when the temperature will be increased, then only they will synthesize here. So, so this is a kind of regulation, right? Where the heat is the principal factor which is regulating the expression. If there is heat, there is expression. If there is no heat, then there is no expression. So it is composed by two consensus sequence, right? Here you can see, and this binding of RNA polymerase to heat shock protein promoter is mediated by a specialized alpha, sigma subunit, as we saw in our last session, that there is a sigma factor. In prokaryote, there is a sigma factor which will actually dictate the RNA polymerase to find their binding site in the promoter, right? Without sigma factor, RNA polymerase cannot be recruited to the chromosome. So for different genes there will be different sigma factors, so that RNA polymerase can be destined, can be recruited to different gene promoters, right? So for heat shock genes, there is a specialized sigma subunit of the polymerase that is called sigma 32, right? So this sigma 32 helps bacterial RNA polymerase to be recruited to the promoter of heat shock genes. So now this is the pattern of two different regulation, negative and positive regulation for the transcription. Right. So now as we are discussing in the context of the prokaryotic cell, then so in the prokaryotes there is a promoter and there is a overlapping site of operator. Right. When there is an operon system, for example, if there are multiple genes are governed by a single promoter, that system in prokaryote is called as operon. So in those cases, there is a specified sequence called operator is present in the promoter region, right? So when and this operator region is blocked or by repressed by some molecule or protein. So when the cell need to produce this so when the cell needs to express that particular gene, right? If you want the transcription of that gene, normally it will not be happen, because the promoter site there is a region called operator, and this operator already a repressor is bound, right? So now as the repressor is bound to this operator, so it is blocking the RNA polymerase access to the promoter, correct? Because there is no place to, or there is no platform for the RNA polymerase so that it can be recruited, right? Because of this repressor here. But when there is a need of expression of that particular gene, then there will be a molecule which is called a molecular signaling molecule that will cause dissociation of this repressor protein from DNA, right? So when the signal molecule will come and then it will bind to this repressor protein, so that the repressor protein will be dissociated from this operator region. So now the promoter region is free, accessible. So and in this situation, the RNA polymerase can come and it can sit over the promoter and it can induce the expression of that particular gene, right? So this is called negative regulation, where the repressor is being bound to the operator region, so that it is blocking the expression of the downstream gene, right? So and the opposite thing is positive regulation, right? So where the bound activator facilitates the transcription process. So again here this positive regulation also, it can have a operator region, right, in the promoter. So here a positive regulator or positive activator, they can bind to the just upstream of the promoter, right, and upstream of the operator, so that it can help to recruit the RNA polymerase, right? So if this activator will be there, then only, if the activator is bound here, then only the RNA polymerase can be recruited. But if you use a signal molecule which will bind and which will dissociate this activator from this promoter region, then what will happen? Now RNA polymerase cannot be recruited when the activator is absent, right? In that case, there will be no transcription, so no expression of that particular gene. So this is called the positive regulation, where this activator is playing role uh, of active of inducer of this particular transcription, right? So these are the two kind of regulation, negative and positive regulation, which we talked about. And then another type of regulation is molecular signal, which causes binding of regulatory protein to DNA. Right? So in this case again, yeah. So when this is a negative regulation, then the operator region in the promoter that is bound by a repressor, right? And this repressor is attached with a molecular signaling molecule, right? But when the signaling molecule will be dissociated from here, right? Then also this repressor will also be dissociated, right? So in that case, when that will happen, then in the absence of the repressor, RNA polymerase will sit over there and it will induce the expression of this gene. So here this molecular signal that is causing the inhibition for the transcription of this particular gene, right? So when you remove this molecular signal, then it will automatically remove the repressor also, because repressor can be bound to the operator only when the signaling molecule is present. When it will be gone, then the repressor will also be gone, and then it will induce the expression of the downstream gene. Okay. So here this molecular signal is causing binding of regulatory protein to DNA. And then if we talk about the, in the context of the positive regulation, then we will see that again in this case, activator is bound, right? Activator is bound in the upstream of the promoter, and this activator is actually associating with the signaling molecule. So when the signaling molecule is gone, then it will cause the dissociation of the activator also. So when the activator is not present, there will be no transcription, right? So here also the molecular signal is playing a role, which is regulating the binding of regulatory protein to DNA. For the left side case, it is regulating the binding of the repressor, and in the right side case, it is regulating the binding of the activator, right? So when it is by helping the binding of activator, so it is required for the transcription. And for the left side case, when this molecular signal will be dissociated, then only the expression will be occurred, right? So in that case, it is preventing. So it is acting as a negative regulator, right? So these are the two different mode of regulation, negative and positive. So where in the upper panel you can see the molecular signal is causing dissociation of the regulatory protein, right? When the molecular signal is getting bound here, then only the regulatory protein is dissociated. Same here, right? For the activation also. But in this case, just opposite, molecular signal is causing the binding of the regulatory protein. So when the molecular signal is gone, those regulatory proteins will also be gone, right? So these are the two opposite way of negative and positive regulation. Okay. I hope it is clear. If you have any question, you can ask. Okay, so next we will see. So till now we just uh, talked about some mechanisms which are imparting the positive and negative regulation for the transcription. So now we will see some example, right, in the prokaryotic only. So in so in prokaryotes, many genes are clustered and their expression is governed by only one promoter, right? So this cluster of genes, when their expression is governed by one promoter, this system is called as operon system. So, for example, in E. coli, there is an operon system called lac operon system because that operon system is depending, the expression of that operon system is depending on the molecule called lactose, right? So, so how the lactose is digested in our cell? So when the lactose enters into the cell, lactose is composed of glucose and galactose, right? So, so now the cell has to express beta galactosidase. So this beta galactosidase will cleave this beta 1,4 lactosidic linkage of the of the lactose, so that it will cleave the lactose into glucose and galactose, right? So now this glucose can be used by the cell, uh, by the prokaryotic cell, which can enter into the glycolysis and it can, produce ATP, right? So in order to enter into the glycolysis, the lactose has to be splitted, and this process is catalyzed by beta galactosidase, right? So this lac operon, there are some genes are clustered in the lac operon. So among all the genes, there is a gene called beta galactosidase. So expression of the beta galactosidase is depending on the lactose only. So now we will see how lactose will govern the expression of the beta galactosidase in the lac operon. So this is the construct of any operon where the there are cluster of three genes here A, B, C. So their expression is governed by this promoter, and the promoter there is a repressor or activator binding site which is called operator, right? And in the upstream of the promoter there is a activator binding site, right? That we saw here that repressor binds to the operator which is lying into the promoter, and activator binds to the upstream of the promoter, right, which is distant, right? Same here. So this is a construct of any operon, right? First activator binding site, then promoter, operator, and then the cluster of genes. So for example here, this is your lac operon. So there are cluster of genes here, right? For example, P1, P is the promoter, right? And then there are two operators O1 and O3, which is followed by three genes Z, Y, and A, right? So the lac Z that encodes the beta galactosidase, lac Y encodes galactoside permease, and lac A encodes thiogalactoside transacetylase. So all of them are important for this lactose metabolism, right? So here the thing is the lac repressor, which is a protein that bind to this O1, which is operator region here in the promoter, right? So operator and the promoter, those sides are overlapping here. So lac repressor bind to the O1 operator so that it can block the access of RNA polymerase. When the repressor is bound, as we saw, RNA polymerase cannot come and bind, right? So expression will not be there. But when we will provide that signal molecule which we discussed, so for here that signal molecule is the lactose. So when we provide the lactose, then this lactose will bind and it will remove the lac repressor from here. Right. So when the lactose will bind to this lac repressor, they cannot bind to the operator region. Right. So on that case, when there is no repressor in the operator region, So in that case, this Z, Y, and A, those genes can be expressed, right? But when there will be no lactose, then lac repressor can bind to the operator region, and then those genes will not be expressed. So that's how the expression system is controlled in the lactose operant system of prokaryotes, right? So now we saw that how lactose controls the activation of the lactopera, right? So, but the thing is that is not only governed by the lactose. There is a protein called CRP, right? Which is uh, right, yeah, CRP, which is the catabolite repression protein. So, or yeah, so which one is cyclic AMP receptor protein or CRP. So that is a which is also known as CAP also or CAP or CRP, which is a homodimer with binding sites for DNA and cyclic AMP, right? And this binding is mediated by a helix turn helix motif within the protein's DNA binding domain, right? So this is your Lacoperon construct. So as we know that in the prokaryote there is operator, there is promoter, and in the promoter there is minus 10 and minus 35 region which is bound by the RNA polymerase, right? Minus 35 is TTGACA and minus 10 is TATGTT, and yeah, so this promoter along with operator region is the binding site for the RNA polymerase. So normally in this operator region, the lac repressor comes and binds, right? So that's why the RNA polymerase cannot bind to the to this promoter region when there is no lactose. When there is no lactose, then the repressor will occupy this place, which is called operator. So then RNA polymerase cannot bind. So then the expression will be switched on. But when this repressor will be removed, then only the RNA polymerase will bind and the expression will be continued, right? And in the upstream, there is a CRP site, which is as we saw, cyclic AMP receptor protein, right? So now we'll see how this CRP site is helping to regulate this lactose, right? So, so for that we'll see the combined effect of glucose and lactose on expression of the lactose. So when the glucose level is low, right, then we know that when the glucose level is low, from our previous discussion we know that the glucagon hormone will be secreted, and this glucagon hormone will activate a signaling cascade, right? So that where the cyclic AMP protein level will be raised, right? Why? Because cyclic AMP is synthesized by the adenylyl cyclase, right? So which used ATP as a substrate and it will make cyclic AMP from the ATP. So this glucagon protein that this glucagon hormone that is a signal for a GPCR, and this GPCR will activate a associated G protein, and as you know that the G protein has alpha, beta and gamma subunit. So when the G alpha will be uh, associated with the GTP, then it will be dissociated from the G beta gamma, and that can only happen when the glucagon will bind to this bind to its receptor GPCR. So then only the activated G protein will activate the uh, adenylyl cyclase, which will produce high level of cyclic AMP from ATP. So that's why in the low glucose level where the glucagon hormone will be secreted, so then you will get high level of cyclic AMP. Just opposite. So in that case, the cyclic AMP will come and it will bind to the CRP, right? And CRP, as we saw that in the lacapanone, that is upstream of the promoter, the region is CRP site. That is the binding site for CRP where CRP binds, right? In the upstream of the promoter, there CRP is binding. But when there is low glucose, then high cyclic AMP level will be high. Then cyclic AMP will come and it will associate it to the cyclic AMP receptor protein, right? It will interact with the CRP. Then, so then what will happen? It will help to express, or it will help to recruit the RNA polymerase to the promoter, right? And then it will get the expression of the downstream genes, right? It will, right? And along with that, when the glucose level is low, then CRP is bound with the cyclic AMP, and when we add lactose, then lactose, and when the lactose is provided and the glucose level is low, then lactose will, as we saw that, it will bind to the, it will bind to the lac repressor and it will ensure the dissociation of the lac repressor from the operator region, right? So now RNA polymerase can sit here because lac repressor is not bound here, but here you can see when the lactose is not provided, then although the glucose level is low, but still the lac repressor is bound with the operator region. So when the lactose will be provided, this repressor will be gone. So now RNA polymerase can bind to the promoter, right? So now in this case only the lac operon can be switched on, right? Now it will induce the expression of the downstream genes, right? But when the glucose level will be high, right, then if the glucose level will be high, then glucagon will not be secreted. So in that case, cyclic AMP, as we saw that, cyclic AMP level will be low, right? Because the G protein will not be activated, and then the adenylyl cyclase will not be activated, so cyclic AMP level will be low. So in that case, this in the CRP side there will be no CRP, because the cyclic AMP, when it will bind to the CRP, then only it can bind to the CRP binding site, which is upstream to the promoter. But when there is no cyclic AMP, then the CRP by its own cannot bind to the CRP site, right? So now in that case, so CRP site will be vacant. So even if you will give the lactose, then of course the lactose will come and lactose will bind to this repressor, and it will actually remove the dissociate the repressor. It will dissociate the lac repressor from the promoter region, right? But still there will be no expression, because in the activator site, right? In that case, CRP is the activator, right? This is a activator for this lac operon. So after adding lactose, although the lac repressor is dissociated, but still there is no activator, there is no CRP, right? So although you are providing lactose, but due to absence of the CRP, which is actually because of the absence of the cyclic AMP, so as there is no CRP or the activator, then although the repressor is dissociated, but still the expression will not be restored. Right? There will be no expression. So for expression of this lac operon, you need to provide lactose, and also you need to keep the glucose level low, right? So that the cyclic AMP level will be high and it can recruit the activated CRP, right? So here you can see you are adding lactose, you are dissociating the lac repressor, but still the expression is not coming there because due to the high glucose, the cyclic AMP level is low, so it is failing to recruit the activator, which is CRP, right? So even if you are providing lactose but the glucose level is also high, then this operon system will not be switched on. But when lactose will be provided and the glucose level will be low, then only this operon system will be switched on, right? So this is called a combined effect of the glucose and lactose on expression of the E. coli lac operon. Right, similar to lac operon, there is another operon system in E. coli, that is strep operon. Right. So this is just the opposite thing, because in lac operon, the lactose molecule was used as a inducer of the expression, right? But in this case, strep will be used as a inhibitor for the expression. So again, if we see the construction of the strep operon. Then we can see that there is a promoter region which is overlapping with the operator region, right, which is followed by a leader sequence or triple, right, and this triple leader sequence has a attenuator site, right, where the transcription will be attenuated or stopped, or halted actually, which is followed by the 5 genes which are the in the biosynthetic pathway of tryptophan biosynthesis. So those genes are ChIP-E, ChIP-D, ChIP-C, ChIP-P, and ChIP-A. So till triple region is the regulatory region, and these 5 genes are the regulated genes, right? So now let's see how this operator system works. So this ChIP-R region, which is upstream of this gene cluster. So ChIP-R region, it encodes a protein called ChIP repressor, right? This ChIP repressor, when it will bind to the tryptophan, it can, so when the ChIP repressor is bound to the tryptophan, then it will bind to the promoter region, actually the operator region of the promoter, which will obviously cause the transcriptional inhibition, because now it is occupying the RNA polymerase binding site. So RNA polymerase will not be recruited, so no expression. Right? So when the tryptophan level is high, then this ChIP repressor will get access of the tryptophan, and this will bind to the promoter region, right? So then this gene cluster expression will be switched off. So tryptophan, when the tryptophan level is high, then the transcription level will be low, right? So it is inhibiting the transcription. When the tryptophan level is low, right? When the tryptophan level is low, then the ChIP repressor is not getting access of the tryptophan, and without the tryptophan, the ChIP repressor cannot bind to the promoter region, right? So if there is no tryptophan, then the ChIP repressor cannot bind to the promoter system. So now in that case, the this ChIP-E-DCBA expression will be increased, right? But when the tryptophan is there, then the ChIP repressor will bind to the promoter region, and that will only allow the expression of the ChIP-L, which is the leader sequence, but these 5 genes will not be expressed, right? So this is the concept of ChIP operon, where the tryptophan is that will be used as a inhibitor for the transcription of this ChIP operon system. So in the ChIP operon system, you can see the ChIP leader, which is a leader sequence. It has a attenuator region, right? So it will attenuate the transcriptional the transcription process, right? So now we will see how this attenuated sequence in the leader triple, that how does it work, right? So this is your mRNA, which is actually the corresponding matured mRNA for the leader peptide, right? So here there are four exon system, 1, 2, 3, 4. After the fourth exon, there is a site of transcriptional attenuation, right? Right, and that is at the number of 139 nucleotide, and the leader sequence end at the position of 162. Right, after that chip E will be started synthesis. So after this fourth exon at the site of 139, there is a transcriptional attenuation site. Okay, so now if you can see that when the chip L, which is the chip leader sequence, when this will encode this mRNA, right? So as we know that in the that after fourth region there is a attenuation sequence. So due to this attenuation sequence, this 3 and 4 exon, they will make a hairpin loop here, right, which is followed by this polyuracil, right? So this hairpin loop made by this 3 and 4 will be actually play a role of attenuator, right, which will actually allow to dissociate the RNA polymerase, so which is basically inactivating the transcription, right? So this is the leader peptide mRNA, and this mRNA is used for the translation of the leader peptide chain, right, by ribosome, right? But when so when this 3 and 4 is making this attenuator structure, then RNA polymerase will be dissociated, right, so that the transcription will be halted, right? But when the 2 and 3 will make a pair, 2 and 3 will make a hairpin loop, then you can see that this hairpin is weak, right? So this hairpin formation, this attenuator formation, cannot dissociate the RNA polymerase. So RNA polymerase will be there, and it will continue the synthesis. It will continue the transcription of the downstream genes, which are TRPE-DCBA. But in this case, when 3 and 4 are making this hairpin loop, then RNA polymerase will be dissociated, right? So the TRPL mRNA will be there, TRPL peptide will be there, but after that RNA polymerase will be dissociated. So the downstream genes TRPE-DCBA, those will not be transcribed, right? So it will inhibit the transcription of the operon system, right? So it happens when the tryptophan levels are high, then the ribosome quickly translates sequence 1 and blocks sequence 2, right, before sequence 3 is transcribed. So now when tryptophan is there, then so as this ORF1 has the chip codons, so when the tryptophan is there, this translation is very fast, right? It can quickly translate the ORF1, right, and it will block the ORF2 before sequence 3 is transcribed, right? So the continued transcription leads to the attenuation at the termination sequence formed by the sequence 3 and 4, right, which is the transcriptional terminator site, right? So when the RNA polymerase encounter this termination site, then it will be dissociated from the mRNA. So that's why the downstream genes are not getting expressed. So now here also the speed is important. So the speed is controlled by the tryptophan level. Because this ORF1 has a chip codon, so if the tryptophan level is high, so the translation will be quickly here. This leader peptide will be synthesized very quickly, right? So now the translation will be going on, and it will block the 2 and 3. So this pair will be blocked, right? Because translation is fast, so it will block this 2-3 pair, right? But this continued transcription will lead to the the attenuation at the 3 and 4, right? If this 2-3 pair is not formed, if the 2-3 pair is not formed, then it will make a termination attenuator structure which is 3-4, right? So this 3-4 attenuator structure will actually lead to the RNA polymerase dissociation, right? So transcription will be halted, transcription will be inactivated. If so, so that's why the high tryptophan level will inhibit the expression of the downstream genes, because uh, because on that case translation will be fast if the tryptophan is there. So translation will be fast. So 2 and 3, before pairing of these 2-3, right? The before pairing of the 2-3, because of this ribosome will be here between 2 and 3, right? Because ribosome is quickly moving here. So 2 and 3 will be occupied by the ribosome, right? Because it is quickly moving. So this 2-3 pair cannot be formed, right? Because of this ribosomal axis. And if that happens, if 2-3 pair cannot be formed, then the 3-4 will be formed, which is the attenuator. And that will leads to the RNA polymerase dissociation, right? So downstream gene will not be expressed. But when there is a tryptophan level is low, then this chip codons in the ORF1, they will actually then the transduction will be slow, because tryptophan level is low. So that's why the corresponding tRNAs will not get their tryptophan. So the translation level will be very slow here, and ribosome will be paused here, right? So ribosome will be paused at the ORF1 where the chip codons are there. So now in that case, so as the ribosome is paused here, so 2-3 is already transcribed by that time. So 2-3 now can be paired, because ribosome is not coming between them, right? Ribosome is not coming between them because ribosome is paused here in the ORF1. So now 2-3 are free to make this pair, right? And as they can make this pair, so that is not a termination sequence. So that's why the RNA polymerase can still stay here, and it can continue the transcription of the downstream genes, right, which are chip E, D, C, B, A. That's how high level of tryptophan is inhibiting the expression of so that's how the high level of tryptophan is allowing this formation of the attenuator structure 3-4, that is inhibiting the transcription of the chip opponent system. But the low level of tryptophan is pausing ribosome in the ORF1, so that the 2-3 are able to pair, right? So as a result of this, 3-4 pairing is not formed, right? So the attenuator structure is not formed here. So that's why, because of this lack of the attenuation structure, the RNA polymerase is not dissociated. It is continuing the transcription, and it is continuing the transcription of the downstream genes, right? So that's why the low tryptophan level is switching on the chip opponent system, but the high tryptophan level, it is switching off the chip opponent system, right? And there is a SOS response in the E. coli, which is a response when there is DNA damage. Right? So when DNA is extensively damaged by UV light, then the DNA replication is halted, and the number of single-stranded gaps in the DNA increases, right? So replication fork will be halted here, stalled here, and that will induce the accumulation of the single-stranded DNA, right? So damage to DNA produces single-stranded gap, right? So now in order to continue the replication, because fork is stalled here, so now if there is prolonged stalled replication fork and there is prolonged accumulation of the single-stranded DNA, then the cell will recruit the REC-A protein. REC-A protein will bind to this damaged single-stranded DNA, right here, and then which will activate the protein's co-protease activity. While bind to the DNA, REC-A protein facilitates the cleavage and inactivation of the LEX-A repressor, right? So this LEC-A, when it is bind to the ssDNA region, then it will be activated and it will cleave the So it will cleave and inactivate the lex A repressor, right? So when the when the repressor is cleaved and the and inactivated, then it will induce the expression of the SOS genes, right? For example, uh, DINE-F, UVRA, POL-B, DINE-B, UVRB, right? SULA, UMUC, UMUD, right? So those genes will be expressed, which are the SOS genes, right? Including the RECA genes, right? So that it can bypass the replication fork from here, right? So again, the RECA bind to the prolonged stalled single stranded DNA, which is activating its protease activity so that it can cleave and inactivate the Lex-A repressor. Lex-A repressor is actually bound to the promoter region of all of these SOS genes. So normally, when the Lex-A is bound with the promoter region of this SOS gene, then those SOS genes cannot be expressed. But when the REC-A will be activated and it will be cleaved, the Lex-A repressor, then the Lex-A repressor will be inactivated. So now the SOS genes can be expressed and they will be helped to bypass the replication fork, right? So that is the SOS response when there is a DNA damage by in the E. coli. So after that we will see the regulation of the flagellin genes in Salmonella, right? So in the Salmonella there are a cluster of genes like HIN, FLIB, FLIA. So this HIN mRNA which will encode a protein which is called HIN recombinase, and Fleece B, Fleece B and Fleece A mRNA, they will encode the Fleece B flagellin and Fleece A protein. This Fleece A protein is a repressor, right? So yeah, so what happened? This HIN is the transposed element. It has come from some somewhere else, right? So that's how uh, the product of genes FLEC and FLEB are different flagellin. This HIN gene encode a recombinase which can catalyze inversion of the DNA segment containing the FLEC B promoter and HIN gene. These recombination site inverted repeats are called Hicks, right? And in one variation, FLEC B is expressed along with the repressor proteins that repress the transcription of FLEC C protein, right? So that's how they can regulate the expression of the FLEC protein, which is FLEC mRNA, which will encode the FLEC flagellin protein, right? So till now we have discussed about the regulation of gene expression in prokaryote. So after that we will discuss the regulation of the gene expression in the uh, eukaryote, right? So in the eukaryote, chromatin, as I told you earlier, chromatin is remodeled by acetylation and nucleosomal displacement, right? So there is along with the chromosome there is the histone protein, right, which is bound with the chromosome that actually uh, so there is some histone modifications which is actually playing some roles to regulate the transcription, right? Right. So many eukaryotic promoters are positively regulated and many are negatively uh, regulated. So this is a typical eukaryotic promoter and regulatory proteins, right? But in this case, for eukaryote, so the genes they have a promoter in the upstream. Along with that, they also have the a sequence where the transcriptional activators can bind, which is UAS, and there is some sequence which are present in very distant from the promoter, that is enhancers. Which is the binding site of the activator, right? Activators are the DNA binding. Yeah, so these activators will bind to this enhancer and they will uh, initiate the or they will induce the corresponding gene expression, right? So before going there, we will first check this, yeah. So that we will start on tomorrow. That the eukaryotic gene expression regulation, so, right? So then our this gene expression regulation will be chapter will be over. Right. So if you see that if we till continue the discussion in the prokaryote, then there is a translational feedback in some ribosomal protein operons, right? Dark proteins that act as the translational repressors are shaded at pink, right? For example, L10S7, they are the L10 means this is the uh, 10th subunit uh, of the large uh, ribosomal subunit. S7 is the part of the small ribosomal subunit. S for S means small, L means large. So the each translational repressor blocks the translation of all genes in that operand by binding to the indicated site on the mRNA. Right? Genes that encode the the subunit of the RNA polymerase are shaded yellow. Genes that encode the elongation. So this yellow, yeah. So this yellow shaded genes are encoding the subunit of the RNA polymerase, and genes that encodes the elongation factor, these are blue, EFG and EF2. We talked about that, right? And R proteins are the large ribosomal subunit proteins are designated to L1 to 34 and smalls are S1 to S21, right? So there are many operons, beta operon. So they are actually this beta beta dash are uh, the these are the subunit of RNA polymerase, right? So this and EFG, EF2, they are the subunit of, they are the actually elongation factor. So S7 is the repressor that, for example, here L10 is the repressor of the translation of this beta operon, right? Beta and beta dash is the uh, subunit of the RNA polymerase. They encode the subunit of the RNA polymerase. S7 is the repressor of this EFG and EF2, which is elongation factor. Alpha operon, the S4 is the repressor, right? Which actually block the translation of alpha, which is the subunit of the RNA polymerase, right? And there is also SPC operon, where S8 is the repressor, and L4 is the repressor for the S10 operon. So there is a stringent response in E. coli, where this response to amino acid starvation is triggered by binding of an uncharged tRNA in the ribosomal A site here, and right, and a protein called A protein called stringent factor will bind to the ribosome and catalyze the synthesis of the triple phosphate G double phosphate, right? So here this is the stringent factor or Rayleigh protein, right? So when there is amino acid depletion or amino acid starvation in the E. coli, then in the A site an empty tRNA will be bind, then this stringent response will be triggered, and the stringent factor or Rayleigh that will bind to the ribosome. Here this is the Rayleigh that will bind to the ribosome, right, and it will be catalyzes the synthesis of the triple PGP. So it will take GTP and ATP as a substrate, and it will make triple phosphate guanosine double phosphate plus AMP. This triple PGPP will be converted to the phosphohydrolase by phosphorylase to this external phosphate will be removed by the phosphohydrolase, which will be converted into PPGPP, right? This PPGPP reduces the transcription of some genes and increases that of others, in part by binding to the beta subunit of the RNA polymerase. So it can bind to the beta subunit of the RNA polymerase, and by binding it can induce transcription of several genes, and it can reduces transcription of the other genes, right? So when it will bind to the beta subunit of the RNA polymerase, then it will modulate its specificity. So some can be so that some genes can be expressed, but some gene cannot be expressed, right? So that's how it can alter the enzyme's promoter specificity. When PPGPP will bind to the beta subunit, then it can recognize some of the promoter, but it cannot recognize the other promoter. So some genes will be expressed and some genes will be not. So that is called the altering of the enzyme's promoter specificity, right? And the synthesis of the ribosomal. So such an example is the ribosomal RNA. Synthesis of the ribosomal RNA is reduced when PPGPP level increased, right? So that will actually eventually inhibit the ribosomal biogenesis. So again, when there is amino acid starvation, then a empty tRNA will be bind to the A site of the ribosome, which will trigger the binding of the stringent factor Rayleigh protein, which will take GTP and ATP to make triple PGPP plus AMP, and this external phosphate will be removed by the phosphohydrolase to make PPGPP. This PPGPP will bind to the beta subunit of the RNA polymerase, and that will alter its promoters specificity. So some promoters will be recognized, some genes promoter will not be recognized. For example, the RDNA promoter or the ribosomal RNA promoter will not be recognized when the PPGPP will be bind to the RNA polymerase. So ribosomal RNA synthesis will be reduced, right, when this PPGPP level increases, right. So that's how they can regulate, they can reduce the expression of the ribosomal RNA, right, when there is amino acid starvation. That is called the stringent response in the E. coli, right. Okay, so that's all for our today's session, where we discuss the regulation of the gene expression in the prokaryote. So if you have any question, you can ask me throughout the session which we all discussed.